Welcome to the Mistra SafeChem final webinar. It's a great pleasure for us to be able to present our research from more than 4 years, the results of research from more than 4 years from this program, and warmly welcome. We have a whole day in front of us with an agenda that looks like this. Yes. The morning agenda looks like this. We have 2 sessions this morning. The first one, which we have already started, which is the introduction, followed by the tools for early warning and proactivity. Secondly, after a short break, we will have a session on catalysis for sustainable organic chemistry, plus upcycling of forestry and textile residues. And please note that we have a Q&A session at the end of each a Q&A slot at the end of each session, where you have the possibility to ask questions, and we will try to answer. And the way we do this is you write your questions in the chat, and we will compile and bring up as many questions as possible. Please feel free to ask any questions about the research or about the program. Now, a short introduction to Mistra SafeChem. Safe and efficient chemistry by design, we call that. What is Mistra SafeChem? It's a research program focusing on green chemistry with a holistic approach to chemicals. How do we do this? By working on new synthesis processes, tools for improved risk assessment and life cycle management, and the sciences hope to predict and avoid risk already in the design phase of new chemicals and new materials. The results of the programs is intended and should reach both current and future chemistry. We are a consortium led by IVL, Swedish Environmental Research Institute, with 5 other research partners and 13 industry partners. The budget is 70 million crowns, it's about 6.7 million euros from Mistra, the Foundation for Strategic Environmental Research, and it runs for 4 years and 4.5 years now, and it's Mistra's largest initiative to date. In addition to the Mistra funding, the partners contribute with an additional 30 million Swedish crowns with both cash and in kind. The program was granted in 2019, started in 2020, and we are right now in the final month of the program's phase 1. And of course, we have a website where you can see the address down there. If you want to find out more about the program, we have our reports, and many of our results that you will hear about today can be found in publications and technical reports on our website. A few words about who the partners are. It's led by IVL, and the other research partners are ChemSec, the DTU in Denmark, KTH here in Stockholm, Rice Institutes, and Stockholm University, who are participating with several groups. The industry partners, they range from, as you can see, pharmaceutical companies to basic producers of chemicals, to smaller and bigger companies that are active along the value chains, of different value chains, but have an active interest in safe and green chemistry. I am very grateful for both to our research partners and to our industry partners who have actively participated in the program. When we started with the proposal and the project, we defined a vision for Mistra SafeChem to enable and promote the expansion of a safe, sustainable and green chemical industry in Sweden. But as you noted from the partners, several partners also are international companies, and also one of our research partners is outside Sweden. So it really has a European and global perspective. The background to the project is, of course, the call for proposals that was prepared and sent out by Mistra, which was to a large extent based on the 12 principles of green chemistry, which have been around for some time, first presented by Professor Paul Anastas and John Warner. And they are still, I think, an important guideline for how the chemical industry should think and how our use of chemicals in society should be managed. It has, of course, been developed in newer versions since the original concept. But more interesting for us is that in 2020, the European Union launched the Chemical Strategy for Sustainability. And this is a strategy that both wants to safeguard people, humans and ecosystems from exposure to potentially hazardous compounds, but also make sure that the chemical and material industry provides the new foundation for the Green Deal, the transition into a climate neutral society, etc. We found after looking at the chemical strategy that it made our program possibly even more relevant and timely, and perhaps especially with the concept of safe and sustainable by design, which is under development in Europe right now, and which also gives a life cycle perspective on safety, sustainability of chemicals and materials. Early on in the program, we made this graphics showing our view of what do we mean by safe and sustainable and green chemistry, and where is our research focused. Here we have as a starting point the renewable carbon feedstock in combination with the design for safety, sustainability and recycling. A large part of the program is focused on this efficient and safe synthesis, providing materials and chemical building blocks, and also industrial and consumer products. We also have research activities on reuse and recycling, but I also want to emphasize these two heading and the lower words here, the life cycle management, the testing and screening. Here we have made large efforts to develop methods that can be used both in the design phase and also in the synthesis and when producing new materials and chemicals. You will hear much more about this during the rest of the day. Now, I have the opportunity to summarize our achievements in phase one in this one slide. Moving from left to right, we see we have engaged almost 100 people at our research partners and our industry partners in various parts of the project, some large and some small, but a lot of people have been involved. And again, thank you to all of you who have participated. So far, we have published 85 scientific articles. There are more on the way. This is the main scientific output from the program. We also have a number of technical reports, and that can be found on our website. We've had 6 external program seminars. We have been running 11 case studies. Some of them started during the program. We've had 2 program conferences, and we also formed a research student group. Now, this we thought was important because we have several PhD students funded by the program at different departments and different universities, and we thought it would be of interest to them to connect and discuss how they contribute to the overall vision of the program and to green and sustainable chemistry in general. The biggest green figure in this graphics is we want to emphasize that we have formed a network. We are really trying to work in a multidisciplinary way with chemists, toxicologists, and life cycle assessment expert to do our research. And this we have some experience of this. It is always a challenge to collaborate between different disciplines, but when we succeed, it's extremely rewarding. And you will hear more about this during the day. So just a quick introduction to what we are going to hear today. We have, as I mentioned, the introduction ongoing now. That will continue with, in a couple of minutes, with early warning and proactivity, experimental and computational tools. After that, we have a short break and we move into the second session on catalysis for sustainable organic chemistry and upcycling of forestry and textile residues. We have, after After a short lunch break, we will continue with life cycle based assessment tools, and we will close the day with a presentation of the Mistra SafeChem Toolbox and a few words about the next steps towards the safe and sustainable chemistry. With that, I would like to give the floor to the next speaker, who is Ian Cotgreave from RISE. Please.
Thank you, John. Yes, good morning, everybody. My name's Ian Cotgreave, and I work at the Research Institutes of Sweden, where I have headed a group working with toxicology. So I'm a toxicologist at heart, and I've had the pleasure of leading the work package that is developing tools for early proactivity and screening and providing information to various case studies and data filling. And so I'm going to show you, introduce this session basically where my colleagues will be illustrating the various tools and their applications that we've developed in this toolbox scenario. And I've stolen a couple of pictures here, or a couple of figures from the original application, which basically showed our aspirations in the area of developing toolboxes for hazard identification and also quantification of exposure. And as you can see here, there are some words in silico. This means basically working with computer technologies to build models for prediction. In vitro, where you're working with cells and biological systems to also provide mechanistic information on toxicity. And also there is a suite of bioanalytical and analytical tools, basically identifying and quantifying perhaps unknown chemicals, but also targeted known chemicals. And basically, as you can see from the figure here, the information from the left to the right was to provide data to appraisals that were going to be made in various case studies. And I've just illustrated the case studies here in a tiered approach. And also to use the data in life cycle analysis to make decisions within the project. And basically what you're going to see today is the result of that effort. And I hope to convince you with my colleagues in hand here that we've achieved quite considerable amounts of success here in the development of the toolboxes. And so you'll be hearing examples from Ulf on in silico tools and computational tools that have been developed. And these are extremely popular, of course, and they're getting better and better in the age of AI, which is the popular buzzword at the moment. You'll also hear about some in vitro tools, some cell biological approaches to providing toxicological hazard data. And you'll also be hearing about analytical tools that are there to detect unknown chemicals. Exposome is the word there, buzzword. You'll be hearing from Britt on that. And also the environmental fate of chemicals, which is very important. And from Ioannis, you'll be hearing from him about that. And then also you'll be hearing an example of how these tools have been systematically applied to a particular case study. And as you can see here, the picture at the bottom here, the pictures in the middle here, it's okay to have a hazard, and if you're isolated from the hazard, then it doesn't become a risk. But if you're exposed to that hazard, then there's a risk involved. And this is just a little cartoon to show that, basically. And I just want to point out that also on the left-hand side, you'll see the toolbox that we're going to illustrate here today in this session. But this is part of a larger SafeChem toolbox where life cycle analyses particularly have their own techniques that are fed by data generated from within the work package that we're going to discuss and talk about now. So without further ado, I'd like to give over the floor to Ulf, who will be talking about the computational tools and tricks that we've been developing within the SafeChem program. So Ulf, the floor is yours.
Thank you, Ian. So, hi, my name is Ulf Norinder. I come from the Department of Computer and System Sciences at Stockholm University. And first of all, I want to say that this is done in collaboration with Swapnil Shaban and Ian Cotgreave at RISE, and also Xie Cheng at IVL that have been developing this toolbox together. Just to put things into perspective into life cycle management, you see that this type of techniques that we are trying to promote there in silico hazard warning techniques are present in several places in the life cycle management, like the materials and the chemical building blocks, the reuse and recycling, also the design in general for sustainability and so forth. And this also ties well in with the green chemistry principles to design less hazardous chemical synthesis, safer chemicals and products, and also when they degrade, that they become innocuous and do not affect life and wildlife and other environmental sort of properties. Now, the current SafeChem in silico has a toolbox, contains a variety of computational models. It's altogether about 35 of them, and many of them are endocrine disruptors. But we also have endpoints like the CMR endpoints for carcinogenicity, mutagenicity, and reproductive toxicity, as well as some organ-specific toxicities related to eye corrosion and skin sensitization, for instance, but also then some environmental hazards, biodegradability, bioaccumulation, and persistence, for instance. And the thing is here that these data sets that we've collected are fairly large in many cases for some of these, well over 1,000 chemicals, which are diverse, which means that the models cover a wide chemical range, which of course is important later on when you're trying to predict things. Now, when you open the toolbox, you will have this kind of input window when you at first, and these models are all packed together into a Windows executable, which is nice because the software is that way secure, because all the data is processed on the local machine that you're working on. So nowhere in the code is there a call to web server somewhere where the structures that you're investigating will be deposited, so which could cause you problems later on, perhaps when you're trying to patent things or something. And you input your structures here by either a SMILE structure, which is the little notations you see down here. For instance, this last row of SMILEs corresponds to the molecule you see here to the right. But you can also search for Cas numbers or names, and hopefully the dictionary in this software can pick that up. You can also use batches of SMILEs that you acquire somehow through an Excel file that you can import and process and get predictions from. So there are several ways of interacting or inputting your structures here. Now, when you develop a toolbox, there are some things that you need to have in place, some fundamental concepts that really needs to be in place, and one of those is the applicability domain. I'll come back to that shortly. But there are also some other concepts that I think that should be in place. That is something of an uncertainty estimation, so you know whether your prediction is very certain or more uncertain, which could influence your choice of structures that you want to continue with. But also interpretability. You know, once you have your prediction, it would be nice if you could relate that back to your structure, what to change in the structure and able to mitigate your toxicity or biodegradability or whatever you are investigating at this point. Now, the applicability domain is a fundamental concept that all prediction tools should have, because we are here dealing with empirical models. We have a set of input features, descriptors, which characterize our structures, log P's, molecular weight, hydrogen bonding. And then we have an output target value, for instance, mutagenicity. And we're trying to fit these input descriptors to our output vector by using some sort of statistical model, using machine learning, for instance. And since this is an empirical exercise, we don't have an equation like the Navier-Stokes equation of flow or something. They have a local applicability domain where they can provide us with reliable predictions, and that needs to be sort of defined. And it's very hard to sort of see this applicability domain, but so you have to have a metric for it. But users of this application must be made aware of if their prediction is an extrapolation or not, because models like this should have bad reputation for the right reasons, namely that they're bad models, not that people are trying to predict things way out of what they are designed for. So an applicability domain is a vital fundamental part of a prediction tool that you're sort of distributing to other users. Now, the model development in this tool consists of 3 sets of models developed by Cytiva, Rice, and Stockholm University. Cytiva, where Xi Cheng was a postdoc while developing these models, by Swapna Shawan at Rice and myself at Stockholm University. And we use both traditional machine learning models with chemical descriptors, as I said, log P, molecular weight, hydrogen bonding, charges, and a whole lot more, or what's called descriptor-free deep learning, where the models learn from the actual graph that you see here what features are important for trying to sort of correlate to the target that you're looking at, for instance, mutagenicity. And the model quality of these models that we put forward are validated both internally by cross-validation, but also externally in the project. And for most endpoints, we have 3 models available, and we then perform a consensus majority vote prediction from these 3, also along with a quantified uncertainty. Once you've done your predictions, you will have an output window showing up where you can see your structure, the smiles, and also the endpoints along with a uncertainty estimation. And these outputs then, if you have many of them, can be outputted into an Excel file for you to use later on in your project for developing a new series of structures, or what is the goal of this exercise. Now, I talked before about also on uncertainty, which we covered here, but also about interpretability. And there are several ways of interpreting these models. In this case, we are looking at toxicophore prediction, and the structure that you see on the left is predicted to be mutagenic. But of course, it would be interesting to understand where in the molecule these mutagenicity comes from. And so in the bottom down here, you see this picture of the structure with 4 different toxicophores identified, which then, if you have a similar depiction for your biological activity, for instance, you may realize that some of these positions are not very important for the biological activity. So you can hopefully change them here and mitigate the mutagenicity that seem to appear in this type of structure. This is one way of depicting it. Another way is to move more into a depiction on an atom basis. In this case, the red atoms are the ones that increase activity or toxicity, while the blue ones are those that decrease it. And thus, if you have a structure like on the upper left here, that you realize has a problem with the toxicity, and it's the oxygen atom in this 3-cyclic membered ring, maybe the biological activity does not have the same influence, and there's so that you can change this oxygen atom into something, perhaps a carbon atom, even though cyclopropanes are reactive as well, and that way mitigate this particular toxic endpoint while maintaining your biological activity, for instance. Lately, we've been spending quite some time on developing what's called H-phrase models. And these data was extracted from the ECHA portal, the H-phrases from the research dossiers. And we conducted various machine learning models using graph neural networks, conformal prediction, and also with different molecular fingerprints characterizing the structures. And to our surprise, for these 10 sort of groups of H-phrases that we developed this model for, develop models for, we see that many of them are quite valid, giving correct predictions, and that the consensus models are quite good in that respect. And also at the same time, for these models, as shown before, we can see that toxicity hotspots can be displayed as a result of the models and the predictions you make. So for instance, here I show that the dark bars on the left is these consensus models where we have what's called very high efficiency, that you will get an answer of yes, it will have an H-phrase or will not have an H-phrase, and that you will have in about 90 or 90+% of the cases when you predict a compound, meaning that the models are quite efficient in being able to discriminate between yes and no in this case, being H-phrase or not. And again, you can have these hotspots identifying, in this case, there are they are color green for different toxicities. And again, if you have a similar picture for your biological activity or something, you may realize that you can change some of these without sacrificing biological activity but by mitigating these toxicities that you are afraid of or you certainly don't want to have in your products or compounds that you're going forward with. So, in summary, you can say that the Mistra SafeChem project have developed this in silico toolbox that provides reliable predictions for these 35 different chemical hazard endpoints. The software is user-friendly and safe because it's only doing local execution. The toolbox has been tested and proven useful for chemical hazard screening in the Mistra SafeChem program, and that these H-phrase models that we just talked about covers all human and eco hazard endpoints within the EU regulation, the CLP, with a good predictive reliability. And with that, I will conclude my presentation, and I will then turn the word over to Britt for her presentation. So, please.
Thank you. My name is Britt Rian. I'm from Stockholm University, the Department of Environmental Science, and I will today talk about proactive screening for persistent chemicals in Swedish waters by non-target analysis. I'm very excited to be here today to present some of my doctoral work. So we have, as a Mistra SafeChem tool, we have developed a workflow for comprehensive screening of chemicals in water samples, which we have now applied to almost 400 water samples. You can see in the pictures here are some of our sampling locations. So what we are interested in is if we can find any persistent chemicals in our water samples. So we and our collaborators have collected 382 water samples, which we have now screened with this workflow that we have developed based on high resolution mass spectrometry and computational tools. And then we do comprehensive molecular characterizations of the samples. And since we are interested in persistent chemicals in the water samples, we have applied a novel persistency-based prioritization strategy, which I will come back to. So first, a little bit of background. Over 350,000 chemicals have been registered in global commerce. This includes both chemicals of unknown structures and also some mixtures, and these have the potential to reach the environment, where it can also transform into other chemical structures. And the majority of these chemicals, they have not been monitored in the environment. Therefore, we have a huge knowledge gap. So we actually don't know everything that we are exposed to through the environment. We think that we can accelerate discovery of these chemicals with using non-target analysis, where we collect all the signals that we can detect in the sample, and then we apply computational tools to characterize the chemicals. And we think we can apply non-target analysis for a more proactive monitoring, where we can actually detect new and emerging hazards before they become widespread in the environment. In water samples, we are also particularly concerned about persistent, mobile, or toxic and potentially toxic substances, PMTs. And this is because of their persistent nature and their mobility in water. They can accumulate in water resources, and they can also be hard to remove in water treatment. So we have been working the first years of my PhD. We have been working on developing a method that covers a wide range of chemicals' physical properties. And just to show you an example here, I have plotted 12,000 chemicals that are relevant to water pollution. So you can see that they have quite a big range of both masses and polarity, and also other physical properties. On top here, I've plotted chemicals that have been classified as persistent and mobile, PMTs, which we are particularly interested in. And then the question was, how do you optimize a method for non-target chemicals? We chose to optimize a method based on 220 chemicals in this case, as you can see in this plot here. So this includes a lot of different chemical groups. It includes pharmaceuticals, pesticides, PFASs, personal care products, surfactants, and so on. So when we started developing this method, we first knew that we wanted to combine first a multi-class target analysis, meaning that we would quantify chemicals that we expect to be in the samples, with also a non-target workflow where we look for a known pollution in the samples. Additionally, we wanted to do minimal sample modification because we don't want to add or remove anything from the sample. So therefore, we filtered the samples and then we injected a large volume onto the instrument. So here we first do a concentration step where we apply online solid phase extraction in line with the instrument. Then we separate the sample using liquid chromatography, and then we apply high resolution mass spectrometry detection. So to be able to cover a wide range of chemicals and also have a good method sensitivity, we ended up with two parallel methods. A reversed phase method for semipolar compounds, which was the most comprehensive one. So here we could detect 205 out of the 220 chemicals. And then additionally, we developed a mixed mode method for more polar and anionic compounds, where we then increased the method sensitivity for 47 chemicals, including 5 previously non-detected. So in this plot here, you can see for all of these 210 chemicals that we could detect, you can see the method limits of detection. And then we have colored here according to the method with the highest sensitivity. And the median LOD of the method was at 1 nanogram per liter. So in the end, we can also say that we cover quite a big chemical space, and additionally, we have a good method sensitivity. So now we have applied this workflow, the reverse phase method for now, to 382 water samples. So you can see in the map here, the sampling locations. So we have samples from all over Sweden. We have done a strategic sampling. So we have, for example, groundwater samples from both rural and urban areas. Similar for surface water samples. Here we also have, for example, surface water that we expect to be influenced by wastewater and so on. We have untreated drinking water, raw water, and we have treated drinking water, tap water. We have analyzed stormwater samples from all 4 seasons, and we have analyzed household, influent, and effluent wastewater samples. So when we have analyzed the samples, you might be curious how the output look like. For each signal that we detect in the sample, so we try to collect all the signals that we can with this method. We have one feature illustrated here as a peak. And for each of these features, we have the accurate mass or the mass to charge ratio. We have the retention time from the liquid chromatography, and we have the intensity in all of the samples. Additionally, we fragment this chemical, and then we collect what we call the MS2 data. And this MS2 data is highly valuable for us because it gives us the masses of the fragments of the compound, and it can therefore tell us something about the chemical structure. So we can apply this MS2 spectra, for example, in spectral library matching. So here we run our spectra, illustrated here as the green one, our experimental spectra, in a huge database to see if we can have a match. It's like a fingerprint matching, basically. So you can see in red here is the database spectra that we have a match with in this example. We can also apply in silico tools that can help us predict the chemical structure based on this MS2 data. So how does this look like in real life? When I analyze my data, this is how it looks like in MSDial, which is a software I use to pre-process my data, align all the data. So you can see here, when you have analyzed almost 400 samples, I have over 70,000 features. So each feature here, each point in this plot have its own mass to charge ratio, accurate mass, retention time, MS2 data, and also intensity in all of these samples. Just as a reminder, each chemical can have multiple features, but you can still see that we have a huge data set with a lot of information here. And this is only in positive ionization mode. We also analyze in negative, where we have almost as much information. So, since we have so much data, we need a way to prioritize what we are interested in. Therefore, since we are interested in persistent chemicals, we built this feature-based molecular library based on wastewater persistency. So then we use this library to then screen for persistent chemicals in our other water samples. So what we did was that we compared 7 pairs of wastewater influent and effluent, and then we classified each single feature based on their median peak area effluent influent ratio, as according to the values you can see in the colorful table here. In this plot to the right, you can see all of the features with its classification and also its retention time. And we could see, we were really glad to see that the majority of our features, they are removed through wastewater treatment, meaning that they were not detected in the effluent at all. And the features that we classified as semi-persistent, meaning that they had a median peak area effluent influent ratio of 50 to 80%, and features classified as persistent that had a ratio over 80%. These features were added to the molecular library and then used to screen for in other water samples. For many of these features, we got spectral library matches with Mass Bank Europe. So you can see this example here. The feature here was flagged as persistent through wastewater treatment, and we had a spectral library match. You can see in blue is the experimental spectra, and in red is the spectra from Mass Bank Europe. So here we had a quite good match with the urban biocide terbutrin. It's used in paint and construction materials, for example. We flagged it as persistent, but we could not find any in the REACH database. We did not have available persistency data, but other studies have also reported this chemical as that it's not sufficiently removed in wastewater treatment, also suggesting that it's persistent through wastewater treatment. You can see that we have quite big high detection frequencies also of this in environmental samples. For example, in surface water and stormwater samples, we detected it in over 70% of our samples. In raw water and untreated drinking water, we detected it in 55% of the samples and in 32% of our tap water samples. I would like to show you a lot of detailed examples, but I don't have enough time, so therefore I give you like a bigger overview instead. So this is a subset of the chemicals that we had spectral library matches with that were all flagged as persistent through wastewater treatment and also detected in environmental samples. So this is only from positive ionization mode. I'm still working on the negative ionization mode data. So you can see that we have detected industrial chemicals, pesticides, organophosphate flame retardants, UV filters, and a lot of pharmaceuticals. And to be able to evaluate if our approach is actually relevant to flag persistent chemicals in environmental waters, I looked up in this huge UBA report on PMT assessment of REACH registered substances that have been detected in water samples, if any of these had any PMT or persistency classifications. Some of these have already been classified as persistent and mobile, or very persistent and very mobile. Some have already been classified as potentially persistent and mobile, according to this report. Meanwhile, the majority, we don't have enough data to evaluate if they are persistent and mobile. You can see the organophosphate flame retardant in the middle here was the only match that I've had that have been classified as not persistent and mobile. Meanwhile, other similar structures, organophosphate flame retardants, have been classified as persistent and mobile. Additionally, the majority of these compounds could also be detected in drinking water samples and in groundwater samples, which also backs up that these chemicals might be persistent and mobile in water. So the next steps in our work will be to first get some more information on the persistency of the chemicals that we have annotated to evaluate how well our approach is working. We will quantify over 200 target chemicals, and we will apply in silico tools for molecular networking, for predicting structures and formulas, and so on. And we hope that we can learn from this work, first off, that this discovery-based approach can help us get a better understanding of the chemical burden in the environment and human exposures. And on a more technical perspective, we hope that we can learn how to connect MS2 fragments or chemical substructures to environmental persistency. Do we have any common fragments within this subset of persistent chemicals? And lastly, we hope that we and our SafeChem collaborators can learn from real monitoring data, real environmental data, which chemicals are persistent, which are not persistent, so we can make smarter decisions when we produce chemicals, that we produce more sustainable chemicals, because persistent chemicals are not sustainable. So with that, I would like to say thank you for your attention. I would like to thank my supervisors, the Chemical Exposomics Research Group, and all my collaborators for collecting samples for us. And also I would like to thank Mistra SafeChem for the founding. And then I would like to welcome the next speaker, Ioannis.
Hi everyone, and welcome to this Mistra SafeChem webinar. My name is Ioannis Liagkouridis. I work here at IVL Swedish Environmental Research Institute as a research scientist. And our work within Mistra SafeChem has been focusing on developing an approach for assessing the environmental transformation of chemical alternatives using a combination of in silico tools, biodegradation testing, and suspect screening. But why is environmental degradation and transformation important for chemical safety assessment? We know well that the major problem with persistent chemicals is that they will accumulate in the environment over time, and that will eventually lead to increased exposures. And as a consequence, the risk for adverse effects also increases. But even if a chemical degrades in the environment, it is possible that it may give rise to equally persistent or even more persistent and toxic byproducts. A very characteristic example is the environmental transformation of PFAS precursors, which are not all of them are necessarily persistent or toxic, but they will transform in the environment into the highly persistent and toxic perfluoroalkyl acids. So it comes as no surprise that there is a regulatory requirement under REACH that the information on transformation products should be included in the risk assessment for chemicals that are produced at higher than 100 tons per year. But when it also comes to choosing a safer alternative, one of the core green chemistry principles calls for the design for chemicals that they not only should not persist in the environment, but they also should break down into non-harmful and non-persistent products. So we have developed an approach to include chemical degradation and chemical transformation in the chemical safety assessment or chemical alternative assessment. A first step in this approach is the application of in silico prediction tools to first assess to what extent and how fast a chemical will degrade in the environment, and secondly, what are the potential transformation products. As a second step, we carry out a biodegradation experiment based on the OECD guidelines. And following that, we screen for the transformation products that were predicted during the first stage, the in silico predicted transformation products. And this information then will be used to inform the hazard or risk assessment in a later stage. And with this approach, we help identify safer alternatives that are compliant with regulations by avoiding toxic or persistent transformation products. And this is very important, especially in an early, for early screening purposes, because in that stage, we generally lack information on transformation products and their environmental behavior and safety. So we applied, we tested our developed methodology in a case study involving 3 alternative plasticizers to phthalate esters. We started by applying a number of in silico prediction tools to predict likely or less likely transformation products. In doing so, we considered both abiotic transformation. You can see on your left side the hydroelectric transformation pathway for one of these chemicals, but also microbial transformation pathway, the one we can see in your right hand, because we tried to cover as many relevant, environmentally relevant transformation mechanisms as possible. And all in all, we predicted 17, 14, and 23 transformation products for these 3 chemicals respectively. And those included major transformation products. We did that in a stepwise manner. And following the in silico prediction, we carried out a biodegradation test to explore the dynamics of environmental transformation and also screen for the transformation products that were predicted by in silico tools. So we were able to identify a number of transformation products for those 3 chemicals at various confidence levels. It was quite important that we identified key first and second generation transformation products, and most of those we observed a significant increase in their levels during the experiment, which means that those transformation products were not just a result of background concentrations, but they were actually formed during the experiment. Although it wasn't within the scope of this study, we did a a quick hazard screening on those transformation products, and it seems that some of those, they actually possess hazardous properties. With that, I would like to conclude. Thank you. I remind you there will be a QA session in the end of this session that you can pose your questions, and I would like to invite a next speaker, Josephine Karlsson, to the stage. Thank you.
Hello, my name is Josephine Karlsson, and I am a PhD student from Stockholm University. And today I will present our research titled The Textile Case, Application of Hazard and Exposure Screening Toolbox. And if you have any questions, you can post them in the chat, and I am happy to answer or try my best to answer those later. As you may know, textile can expose us to a variety of chemicals every day when we wear them, and some of them can pose significant health risk. And our research group focus on developing analytical methods, mostly based on mass spectrometry, to study the occurrence and potential health effects of some of those chemicals. So we have employed three main screening strategies, and those are based on target screening, suspect screening, and non-target screening. And in the target screening, we focus on detecting and quantifying target compounds belonging to classes such as nitroanilines, quinoline, dispersed dyes, and so on. Through the suspect screening approach, we are aiming to identify suspected chemicals, and those can be, for instance, dispersed dyes. And this type of class doesn't have so many reference standards available unless you want to buy kilograms of chemicals. And through a non-target screening approach, we aim to identify new chemical classes which we don't know yet or don't suspect to be there. And the analytical method we used for those types of workflows and screening are either gas chromatography coupled to mass spectrometry or liquid chromatograph coupled to mass spectrometry that can either be high resolution or low resolution depending on the chemical's properties. So our screening study has revealed the presence of several nitroaromatic compounds present in textiles, and some of the ones that we detect have been above the levels for the regulated aromatic amines that are sold within the European Union. And importantly, many of the ones that we detect are today not regulated. In the suspect and target screening of dispersed dyes, we could identify 62 dyes, and we could confirm the presence of 11 of those. Interestingly, we saw that dyes that are used in clinics to test people with the suspected textile allergy were not so frequent found in the clothes on the market today. So to sum the screening result, we could detect several dispersed dyes, and many of those can be reduced to mutagenic compounds and or skin sensitizing components. Compound classes such as quinoline, nitroanilines, and halogenated dinitrobenzenes are frequently found in garments, mostly in polyester garment. The non-target screening showed the presence of other classes, for instance, nitrophenols. So to judge from the structure of those classes, it seems that several of those components are added during the dyeing process, and maybe a way to obtain cleaner textile could be to have cleaner dyes. And it is also important to understand what the dermal exposure to those kind of compounds from textiles means. And we have also developed a method in order to increase the knowledge of chemicals in textile, which is based on the direct screening of textiles. And this method uses an instrument which is an automated thermal disruption coupled to a gas chromatography mass spectrometry. And in this method, small pieces of textiles are added into a tube, and the tube is then heated and the vapor is directed to the gas chromatography mass spectrometry for separation and detection. And this kind of method provides a solvent-free and minimized manual sampling handling for the screening of textile, and it can help to support decision when reusing and recycling textiles, and it can serve as a fast screening and control of hazard chemicals in textiles. So we developed the method for more than 30 compounds belonging to classes that we have previously detected, such as quinolines, nitroanilines, dinitroanilines, phthalates, and halogenated dinitrobenzenes. And here you can see an example of how a chromatogram can look from the result. And we have analyzed a garment which a patient were suspected of textile allergy. But so far we have actually not been able to find the hapten that the patient reacted to. So to summarize the developed ATD-GCNS method, this method is suitable for compounds with molecular weight and lipophilicity, which favors migration and skin absorption. It showed sufficient quantification limits when based on the current European Union levels for the study compounds. It provides a solvent-free, automated, and relatively fast. One sample takes approximately 40 minutes, and it does minimize the manual sampling, handling, and also lab consumables. It is proposed as a standard method by SIS to be used within the European Union as a screening method, and we are still awaiting for the decision. So we have also been involved in a collaboration within the Mistra project, where Professor Ajay Matthews and Maria has derived cellulose nanocrystals from mixed mode post-consumer garments. And our involvement has been to screen for organic pollutants in all parts of this process. So we have analyzed the garments itself and also the derived cellulose nanocrystals, the recovered polyester, and aqueous effluent. And the process you will hear more about later from them. And when we look at the comparison of compounds that we can detect in the textile versus this produced cellulose nanocrystals, we visualized it in this plot. And on the x-axis, there is the log fold change. And the more to the left means that the compounds are present at higher levels in the polyester cotton garment, the starting material, and the more to the right, they are more in the cellulose nanocrystals. And also the higher up in the graph, it means that it's more significant. And what we could observe from the screening study, we detected fewer compounds in the cellulose nanocrystals. And we also did quantification of some compounds, which we have the reference standards available. And it is showed the distribution during the process in this figure. And the brown corresponds that the analytes were quantified in the recovered polyester. And the yellow is cellulose nanocrystals. And then the green one is the process effluents. And we can see that depending on the classes it belongs to, they distribute differently. For instance, quinolines and nitroanilines are mostly found in the polyester part. Dyes depend on the classes. For instance, the dispersed dyes distribute to the polyester, while basic dyes follow the effluent. And also we could see that PFOS follow the effluent to 100%. Phthalates, we could actually see that it's accumulated within the cellulose nanocrystals. And it is important to consider this type of classes, but in this case, it's also important to mention that they were well below the current regulation within the European Union. And we have also studied the possible skin uptake and hazardous properties of halogenated aromatics compounds that we quantified and detect in textiles. And we investigated the migration of this compound into artificial sweat. And we could see that those compounds can migrate into sweat to 0.6 to 24% from polyester garments. In a collaboration with RISE, they did the AIMS test, and from those results, they showed that 2 identified textile compounds possessed mutagenic potential, and this add 2 additional mutagens that are frequent present in textiles. In combination with us plus RISE, we studied skin sensitization using OECD-defined approach, where we combined the outcome of 2 different in vitro tests, which were the direct peptide reactivity assay and also the human cell line activation test. And these results show that at least 3 skin sensitizers are present in textiles. We also assessed the risk, what dermal textile exposure would mean. And we saw that by taking only the individual compounds, the risk seems acceptable of textile exposure to these compounds. To summary and conclude, by applying the developed tools and workflows, it revealed the myriads of chemicals in textiles that could be taken up by human skin. Compounds with mutagenic and skin allergenic properties occur in skin clothing. And we could also see that the chemical content is reduced in cellulose nanocrystal compared to the starting material in the study upcycling process. And for the phase 2, we will also continue to further investigate the detected compounds and also include mixtures.